the chair of the biology department and also the associate director of education in CEMB. So I want to welcome everyone here, and hopefully if people also on Zoom. This is a particularly bittersweet year for the CEMB REU program because this is our last iteration of the CEMB REU program. So you guys are ending this on a high note. So thank you for that. What is 100% sweet though is now we get to hear about all the cool stuff that you guys have been doing this summer. So I hope you enjoyed the research experience and got to know people in the lab and broadly in at WashU. Hope you also took time to get to know WashU in St. Louis if you're not from here. Just a few other things I want to mention because just as sort of reminders really. One is we are now forever part of your scientific network. So don't forget that. So if you have questions or have any way we can help, you should feel free to reach out to any one of us in CEMB and your lab. The other is your Bayer mentors are now also part of your scientific network. So don't forget to take advantage of that resource. And that's really a unique resource that you guys have access to. And so I would urge you to continue to sort of take advantage of these, your scientific network here and beyond. I mean, we love to hear back from our students, how you guys are doing, what's happening as you sort of progress in your career. So, send those emails and notifications, and we love to hear from you guys. Lastly, I just want to take a moment to let's thank all of your bench mentors. I know at least I see a couple of you, right? So your bench mentors, as I'm sure you all appreciate, have been sort of really key to your summer research experience. And so really thank you to the bench mentors. Please let's also thank the Bayer mentors, some here. They really take time from their busy days to make time to interact with you guys. And again, I urge you to keep those interactions alive and active. And finally, a big thank you to both Gwen and Patricia who are making all of this happen. So thank you very much for the program. Now I'm going to pass it over to Patricia to get us started. And apologies if you see me sneak out, because I have a review panel that I'm on that I can't miss. So probably leave the mic. Thank you.
Thanks, Ron, for all your help. Welcome, everybody. It's so great to see a nice support group here too, as well as the students. As Ron said, this is our final year. Having the program, you guys are really setting the program off on an excellent note. I think it's one of the best cohorts that we've had. You guys have been supportive of one another. You've been prompt in your participation in all of our activities. You've done some excellent research, and we're very, very proud of you. And we can't wait to see all your work. And I just want to second what Ram said too about all of us being part of your support network now. And that's next week, next month, next year, whenever, right? You are part of this cohort, and it's you can provide support, you can receive support. It's really a strong part of what we do. So what the plan for the next few minutes here, the next couple of hours here is the students will present one by one for about 15 minutes. They will, for the most part, be going through their posters on the board, and we can have some time for questions. We are not in a rush. If we run a little short or a little long, it's okay. But we'll try to keep to time. Is there a break built in, Lynn, or is there? Yeah, so if it seems like we need one, we'll take one in the middle. get caught up. So we are recording if, and welcome to those who are watching us on Zoom today. So let's begin with Ellie Berkland from the Dai Lab.
Yeah.
I'm just trying to full screen it and I can do it on the Windows machine.
You scroll down to the bottom, scroll to the bottom. And then, just kidding.
Right?
Wait.
Like I reached my nose video.
Opened up in a browser.
It's open up in a browser, so you doesn't work that way. It's normal.
Okay, maybe I knows how to work a Windows machine. Thanks. Not a thing.
No.
Thank you. Well, now what did you do? Something
F11.
F11, thank you.
Oh,
you
know, it's okay.
I forgot it. I'll go at it.
No, you're.
Okay.
Okay, hi everyone. My name is Ellie Berkland. I'm a junior studying chemical engineering here at WashU. In the summer, I worked in the Dai Lab. So the Dai Lab focuses a lot on intrinsically disordered proteins, which I will refer to as IDPs for most of the rest of this presentation. But basically, they're proteins that don't have a structure. So normally, for a protein, the function is dictated by the structure. So for IDPs that don't have a structure, it's often difficult to determine what their function is within the cell. And so we have been observing that. And then another important part that we look at is these IDPs can what's called phase separate. Basically, you can think of it as like an oil droplet in surrounding water. And so we refer to those as condensates. It's when the IDP will basically self-associate and separate from the rest of the cell. So my projects focus specifically on directed evolution, which is basically where we take an intrinsically disordered protein and put it through a bunch of mutations to make different copies of the IDP and to change its structure. So our motivation behind this was just to build a library of mutations to try to determine what amino acids are affecting the structure of these IDPs. And the background on this is the Dai Lab had done some other research with IDPs where they used a selection method, a survival selection, where if they put the IDP in a cell along with another protein called protein E, which would cause the cell to self-destruct if the protein was out in the cell. So if the proteins phase separated, if the IDPs phase separated, they would trap protein E inside the condensate and allow the cells to survive, as you can see on that plate. And then from there, they did further testing on those cells, so imaging, looking at the condensate within the cell. My work was relatively similar to this, but we didn't use a selection method. So we took an intrinsically disordered protein that we had on hand and put it through around 7 rounds of error-prone PCR to introduce point mutations within the protein. From there, we took the protein and we transformed it into the E. coli and allowed it to grow. And then afterwards, we ran it under colony PCR. The purpose of colony PCR is to basically ensure that what we're sending to the sequencing company actually contains the DNA and the protein that we can sequence and get the results that we want. So we basically put the E. coli in a solution, a PCR solution, and heat it up to lyse the cell. And then we will run it through a gel electrophoresis to ensure that that band containing the DNA and protein is still present. From there, we could send it off to be sequenced, and then we were able to analyze the structure using different prediction tools. The first thing we did was we obtained a mutation library from the sequencing results. So I don't know if you can see the big blue slice, the key says we took, basically this is an amino acid shift from a hydrophilic amino acid, so an amino acid that loves water, to a hydrophobic amino acid. And we can see that that occurred very commonly within these proteins. And then another example of something that occurred was an N protein shift to a D protein shift after the directed evolution, and that's a hydrophobic to a hydrophilic amino acid shift. So that pie chart shows all the mutations that we observed after 3 rounds of directed evolution. The next thing we did was show this gel electrophoresis. So this is a gel from the colony PCR. And obviously the bands are present, which means that the proteins were also present within the pipe. And then the bottom band on that DNA ladder, it's called, is around 500 base pairs. So we were looking to have around 600, a little bit over the 500 base pairs. And so all of those that are the higher level, the bands of DNA, those are the predicted results that we wanted. So we were able to cut those out, purify them, and then send them off to sequencing. The next thing we did was analyze the sequences that we obtained from the sequencing results. And we used 3 different prediction tools to gather data on the IDPs that we had mutated. The first thing we did was a predicted structure tool to look at the structure of the proteins after they had been mutated. The next tool we used was a disorder score prediction tool, which shows whether the protein remained disordered or. And then the last one is a phase separation prediction tool, which tells us how likely the protein is to still phase separate and form that condensate, as I talked about earlier. So the first prediction tool we used is called AlphaFold. You can see in the center of this, that's the protein that we started with. And you can tell it's disordered because of a couple things. One, oh gosh, now I don't. You can tell it's disordered because obviously there's not really a clear structure. And then also, this is called PLDT. It stands for Predicted Local Distance Difference Test, which is basically a test that the prediction tool would use. It's a per confidence or a per residue confidence score showing how likely the protein is to actually be what the AI predicted it to be. So since this one is very low in the center, we can see that it basically doesn't have a fixed structure, but this is what the prediction tool could come up with. So it's constantly changing, but that gives you an idea. Around the outside of that is all of the different mutations that we were able to obtain after 3 rounds of directed evolution. So you can see this top one side and this other one are an alpha helix, and so they form some structure after the mutation. And then the 2 on the right show a beta sheet mutation, so forming structure after 3 rounds of evolution. And then you can also see there are 2 that still have a very low confidence score, meaning that even after 3 rounds of mutation, they still remained completely disordered despite the amino acids changing. The next thing we looked at was the disorder score. So along the y-axis is the score from 0 to 1. Above 0.5 indicates that the protein remained completely disordered, and below 0.5 means that the protein obtained some order after mutation. So the x-axis shows position, which is basically saying like 1 through the end of the amino acids. Our IDPs that we looked at usually had about 160 amino acids in their protein, but some were cut a little bit short due to like a stop codon. Or something like that. But you can see that on here, it's kind of hard to see, but the original is in blue and it stays completely above the 0.5 line, indicating disorder, as we can see from the structure also. And then there's also a couple that have obtained order after those mutations. Another important thing to highlight is that some of these on here still have a disorder score above 0.5, basically indicating that, as you can see, it's not, the website is not super confident that the structure has occurred, so the disorder score still shows that. Even though we were given some order, it's maybe not quite as ordered as we think. The last thing we looked at was the phase separation, so the ability of the protein to separate out from the rest of the cell and self-associate. And we noticed from past studies and also from this study that if the protein remained disordered, it was more likely to have the ability to phase separate and to self-associate with itself, forming a condensate. And so you can see the lower bands correspond to a more ordered score from the disorder score prediction tool. So in conclusion to this, we were able to build a library of over 51 mutants, and which shows just in a short amount of time how much data that you can collect. And then from this, we hope to take it and use what we did in Protein E and determine how the IDPs affect the cell and its function and also the cell's viability, and also understand which amino acids can affect the structure of the protein and hopefully learn how to harness that to the best. Oh wait, so does anyone have any questions? Go ahead.
Great presentation. I really like how you presented that like story that you took, so like good job on that.
Thank you.
My question is, if you take the best example of these mutated proteins that you find in your library, how many amino acid changes took to order a disordered protein? Like how, like, do you have like a number?
I don't specifically. I know that, so I would say in the first round, we had a little over 20 mutations, and the second and third rounds were closer to 100 mutations each. But that's obviously across 15 proteins. And so this one is actually probably the highest confidence score that we obtained, and that was after the third round. So I don't have a specific number, but it was a decent amount considering the amino acid sequence is only 160 amino acids. Go ahead.
So evolution has been described as survival of the fittest. I was curious, what is trying to survive here? What are they struggling against? What is your selection mechanism?
So in this project, we didn't have a specific selection mechanism. It was basically just to build a mutant library to figure out if we can manipulate these intrinsically disordered proteins. So these proteins, if they phase separate, sometimes they'll lead to cellular diseases such as cancer or Alzheimer's. And so the hope is to kind of figure out how to manipulate this and what is causing the condensate formation, like what is causing the interaction for them to self-associate versus like stay within the cell. If that makes sense. Does that answer your question?
Part of it.
Okay.
I have a second follow-up question.
Yeah.
Which is then how are you measuring phase separation?
So we just used a prediction tool. This basically just tells us like how likely it is to phase separate. But also it's more likely to phase separate if there's large disordered regions, because when I did a little research on it, basically if it's called multivalency, but basically if there's multiple sites that can connect to like one portion, it's more likely to phase separate. So the more disordered, the more likely it is to phase separate. But there's not really like a great way to be like, oh, this is 100% gonna phase separate. Like that's something that's kind of still unknown, especially like we were talking about, why would it phase separate and self-associate versus hydrogen bond to the water or whatever's around it.
And then how would you know that the colonies you selected didn't have some special phase separation behavior?
I think that was honestly the fun part of this project is we didn't really know, like we probably grew hundreds and hundreds of colonies, and then just picked random ones, whatever the mutation was. And so that's why we were able to obtain such a random library, is because we don't really know. And so this was not to say it was preliminary, but it was a very like preliminary study to determine.
Summer, very impressive. 51 funds. I think you'll see some of other work here. We don't get to such a high number, and well, yeah, impressive.
Thank you. Makayla, go ahead.
For the disordered scores, how do why do the proteins go this disordered?
Basically, it's just due to like the amino acid mutation. So you can kind of see, this one is a good example of like you see order here. So there's an alpha helix from the amino acid shifting, but not all of those amino acids shifted. So some of them that remained the same will still remain completely disordered. And so you have ones that dip down and become ordered for a short time, and then it goes back to the original amino acid sequence, or even with mutation, it didn't change the structure, if that makes sense.
Is there like a importance for how long they stay ordered? Like, does that say?
I mean, it just tells you that they're like the alpha helix is longer and that there's more, there must have been a mutation that caused the structure to stay ordered for longer. But not really. It also just kind of depended, like the two that really dip down and then go back up, those are also, they're not a shorter protein, but what is red is a shorter protein because there's a stop codon that was mutated into the protein, causing it to like not read the protein anymore, if that makes sense. So it's more just that there's not a certain time frame that makes it like perfectly ordered, but sometimes it'll just be a shorter sequence.
I was just curious, how common are IDPs versus normally folding proteins? Like, how common or rare are they?
So that's actually a really good question. When I kind of talked, you can correct me if I'm wrong, but Professor Dai did a talk at the start of the semester or the start of the summer, and he said they're fairly common in our cells even, just because we're still trying to figure out the function, but they obviously serve some purpose. So yeah, I can't give you an exact number because I don't.
58%.
Okay, yeah. Yeah, it's fairly, yeah.
My goodness.
Yeah.
Thank you, really cool talk.
Yeah.
And then for the other question, just trying to put it up.
Yeah.
You're right here.
But go ahead.
What she's trying to do is to convert mouse structure to structure to see what's her strength gonna be.
Yeah.
Exactly the relates with exactly. Actually, many of the IDPs, they serve as a soluble, like transcriptional map. They don't need to, some of them just need multi-level interaction. They don't really need to phase out. So, but from an evolution standpoint, she asked a very interesting question, like how disorder to structure the encode function. That is going to be her next step for how this, because she starts from a soluble protein, totally dissolvable protein. Then what's a minimum motif if needed to encode some of the function critical to survival? That will be her next step. So she, I think, finished some pretty amazing.
Any other questions? Thanks so much, Ellie. We have Salama, the role of NEP1, maintaining nuclear isotope integrity, and McNeill Lab.
All right, hello everyone. My name is Ali Salama. I'm a rising junior studying biology at the New Jersey Institute of Technology. And today I'll be talking to you a bit about a protein called NEP1 and its role in maintaining nuclear envelope integrity. But before I begin, let's talk about what the nuclear envelope even is. So the nuclear envelope is a double-layered membrane that surrounds the nucleus and provides a barrier between the nucleus and the surrounding cellular environment. It is worth mentioning, though, that the nuclear envelope is not just a passive wall between the two. It acts as a communication interface, constantly exchanging signals and materials between the nucleus and the cytoplasm. It's also worth mentioning that it's a pretty dynamic structure. Its composition changes during cellular differentiation. It deforms due to mechanical stress. It repairs itself after rupture, and it even fully decomposes and reforms every time the cell divides. Now, this kind of remodeling is very important for cellular differentiation, division, and growth. And defects in this process lead to serious diseases like cancers and muscular dystrophies. It's also this kind of plasticity that allows the nuclear envelope to maintain a lot of mechanical stress. And when cells experience a lot of mechanical stress, they are prone to rupture. And if rupture is not repaired quickly, the genome can be exposed to damage that can be undone. This kind of genome instability is what leads to cancers in the first place. So what drives this nuclear envelope and its dynamic and its integrity? Well, a protein called NEMP1, or nuclear envelope membrane protein 1, is a protein that has been studied by the McNeill Lab and has been shown to be involved in maintaining the integrity of the nuclear envelope. It was found in the McNeill Lab that the NEMP1 is involved in maintaining the stiffness of the nuclear envelope. Without NEMP1, when you knock down NEMP1 in cells, we find that the nuclear envelope is a lot less stiff, and this leads to early onset female infertility. This is because the ovarian cortex is a very mechanically stiff environment, so developing cells in this environment are prone to a lot of mechanical stress, which leads to physical deformation and damage. Now, in our preliminary data in our lab, NEMP1 is also shown to have a role in protecting the cell against nuclear rupture. As you can see in figure 1, with NLS RFP indicated in orange, when you knock down NEMP1, cells are more prone to rupture due to mechanical compression. We did this experiment using a solution called PEG, which I'll talk a bit about later. And NLS RFP here is a reporter for the nucleus. What you have to know about this for now is that when NLS RFP is in the cytoplasm, we know that there's a rupture because the nuclear contents are not supposed to be in the cytoplasm. So as you can see, after 10 minutes, there is a rupture of the nuclear envelope when you knock down NEMP1 compared to control cells. Not only this, but there's also larger nuclear deformation due to this mechanical compression. So we know that NEMP1 is involved in maintaining the integrity of the nuclear envelope, but this raised another important question. Is the abundance of NEMP1 limiting to the cell? In other words, if we increase the expression of NEMP1, will this increase mechanical integrity or will this? So to test this question, we had to come up with a model. And to do so, we overexpressed NEMP1 in RPE1 cells with an NLS reporter. We use RPE1 cells because they're a well-characterized human cell line. It's non-cancerous and it's easy to manipulate and image consistently. We use the NLS reporter again because that allows us to use confocal imagery and see where the nucleus is. To do the overexpression, we use a method called transduction, which is essentially a process by which a virus transfers genetic material from one cell to another. We used 1 microliter of a NEMP1 GFP lentivirus and added that to a well containing 50,000 cells and let it transduce for 2 days. And then we confirmed the overexpression using confocal microscopy with NEMP1 GFP shown in green and NLS RFP shown in orange. And then we calculated the transduction efficiency by basically counting the amount of cells that were overexpressed in NEMP1 GFP. We calculated the transduction efficiency at around 45.9%, which gave us a big enough population to begin our experimentation. But before we could actually test the hypothesis, we needed to test whether our model actually worked. So to do so, we did something called a laser rupture assay. Essentially, what we know about NEMP1 is that it is also involved in repairing of the nuclear envelope. So if you rupture the nuclear envelope, you see a accumulation of NEMP1 at the nuclear envelope rupture site. So theoretically, if our model did work correctly, we would be rupturing the nuclear envelope and NEMP1 should accumulate at the rupture site. So we did so using this laser rupture. What laser rupture allows us to do is induce damage at the nuclear envelope at a precise location that we choose and a precise time point, which allows us to track the repair of the nuclear envelope frame by frame. So we did so in figure 2 using our model, and we found with NEMP1 GFP being shown in green and NLS RFP being shown in orange, by 15 minutes there is a slow accumulation of NEMP1, and by 45 minutes there is a full accumulation of NEMP1, showing that NEMP1 is in fact being involved in the repair of the nuclear envelope as we had expected. So now that we had a working model, we can now move on to actually testing our hypothesis. So we did so using a similar methodology to the knockdown experiment. We did mechanical compression using the 5% PEG solution. Essentially what PEG is, or polyethylene glycol, is a high osmolarity solution, which increases the osmolarity of the cell medium. What this does is cause a rush of water outside the cell, which causes a decrease in volume of the cell and places a lot of stress on internal structures, including the nucleus, which is what we want. So again, with NL's RPP shown in orange and NEMP1 GFP shown in green, in figure 3, you can see that by 10 minutes, there is a slight compression of the nuclear envelope, and by 45 minutes, there is deformation of the nuclear envelope, with some cells even rupturing, although it's not very visible by eye. However, after quantification, we found that the NEMP1 expression does not actually affect nuclear deformation or the rupture frequency after mechanical compression. Now, this was kind of surprising to us. Our hypothesis initially was that overexpressing NEMP1 would lead to a more stiff nuclear envelope and more less susceptibility to nuclear rupture. However, this was not the case, but this information is still informative to our understanding of the role of NEMP1 in maintaining nuclear envelope integrity. Basically, what this pattern shows is that NEMP1 is not the bottleneck in the process, and it could be another bottleneck in maintaining the integrity of the nuclear envelope. What we concluded was that NEMP1 interacts with other proteins to induce the maintenance of the nuclear envelope integrity. And this is further proven by data that we also have showing that LEMD2 interacts with NEMP1 in maintaining the nuclear envelope, specifically with repairing the nuclear envelope after rupture. In this experiment, cells were knocked down of LEMD2 this time, and we tracked NEMP1 during the nuclear envelope rupture. So cells were ruptured again using laser rupture with NEMP1 GFP shown in green here. And as you can see with the rupture site, the cells with knockdown LEMD2 had less of an accumulation of NEMP1. Not only was the intensity of the NEMP1 puncta decreased, but the aspect ratio was also increased, indicating that without LEMD2, NEMP1 still accumulates, but it fails to accumulate in the concentrated fashion that it usually does, which inhibits repair. So zooming out a bit, this information is important because knowing the role of NEMP1 in maintaining the nuclear envelope integrity is not just like a cell biology detail, it's directly related to genome stability, and genome stability is directly related to different diseases like, of course, cancer. So by understanding the role and mapping out the different proteins that are involved in the integrity of the nuclear envelope, we can further understand the process and eventually do something about it. And finally, I would like to acknowledge everyone at the McNeill Lab, especially my mentor, Yuan Hong, and my PI, Dr. McNeill. Thank you all for listening, and I can take any questions.
Is NEMP1 like a repair guy, or is it like a structural component? And when you overexpress NEMP1, even though they like popped at the same frequency, did they fix at like a higher frequency? Like, did they repair the envelope faster?
Yeah, so we tracked both, and actually it was kind of the same thing, but we didn't have enough data to really make a confident statement about that. We more made the statement that it's there's less of an effect in nuclear deformation and rupture frequency. However, NEMP1's role is really dynamic. It is actually conserved across all metazoans, so all animals across the animal kingdom. And what this shows is that it holds like a really important cellular function, because generally speaking, proteins that are conserved across evolution, they have a very important cellular function because any differences to them are not well tolerated in the like evolutionary timeline. So it is involved in the nuclear envelope stiffness, it's involved in the repair, it's involved in like less susceptibility to rupture. And these could all be related, but generally NEMP1 has a very important role in maintaining the integrity, which is more of like a blanket term for all of those dynamic properties.
Great research and presentation, Ali. I'm curious for NIMP1 and LEMD2, how is that interaction determined? Do you think there might be other proteins interacting as well in this process?
Yeah, so this project was kind of a build on my mentor's project, which is overall investigating the role of NEMP1 in nuclear envelope repair. So a lot of the data that my mentor collected in nuclear envelope repair was investigating these different proteins because he's trying to map out the whole process. With NEMP1 being one of the key interactors in the whole complex. There are other proteins, including a protein called fumarate, and of course LEMD2 that are involved in this process in repairing the envelope. While NEMP1 is one of the more late stage repair proteins.
Questions? Thank you, Alex. We have Anisah Dowell with the Bose Lab. Never seen the whole thing at once, but it's okay. It's fine.
Hello everyone, I'm Anisah Dowell. I'm a writing senior here at WashU studying environmental biology. In the summer I was working in the Bose Lab. My project focused on the potential of an iron-oxidizing bacteria, also reducing iron. So as some background, we have Rhodopseudomonas palustris type 1, which is a purple photosynthetic bacteria that is able to uptake electrons through iron oxidation through ABC, a protein complex. So it has also recently been found to be able to grow on oxidized iron as well as positively charged electrodes, which makes it plausible that it could also grow through iron reduction. So iron cycling is critical for microbial survival because iron is only soluble as iron 2, and that's a limiting nutrient often. So Ty1 we know can oxidize iron 2 into iron 3 through PIO-ABC, a protein complex. But it is unknown whether Ty1 can also reverse this process, reducing iron 3 back into iron 2. But if it can, then that'll benefit the surrounding microbial communities because it'll provide iron 2 to those microbes. And it can also be leveraged for bioremediation efforts. Our main questions are, can Ty1 reduce iron 3 and is PIO responsible for this process? So our methods, so we sealed bulk tubes and made them anaerobic using N2CO2 gas, and then we provided them with freshwater media, which met their minimal nutrient needs. And then we added one of 2 iron sources, either iron 3 chloride or iron 2 chloride at varying iron concentrations to see if the amount of iron has any effect on the amount of oxidation or reduction. And we provided a carbon source, 1 millimolar butyrate, and NTA to keep all of the iron soluble. And we measured their growth using or putting the samples as they grew into a spectrometer, measuring at OD660. And the reading revealed the amount of biomass that they were able to accumulate. And we measured the iron as they grew using a ferrozene assay. So we put an aliquot of the sample into a 96-well plate, added HCl and ferrozene, and the intensity of that purple that the sample turned revealed the amount of iron 2 in the sample. So more purple was more iron 2. So moving on to our results, this is set up by iron concentrations. We have 100, 400,000 micromolar of iron. So for the iron 2 chloride growth, we expected that the pio knockout strands would grow less than the wild type, solely because they were not able to take advantage of the iron 2 electrons through oxidation. However, we did not see this to be the case. The iron or the pio and wild type and each iron concentration grew to the same amount compared to each other, which could be because pio ABC is energetically expensive to make. So there is a trade-off. So in the end, they grow the same amount. We did see that the iron 4 concentration grew significantly more than the other concentrations, which could point to iron 2 actually having an inhibitory role on Taiwan growth. In the iron 3 chloride growth curve, we expected that there wouldn't be any difference between the delta pio or the pio knockouts and the wild type streams, because neither of them are able to take advantage of iron oxidation because we didn't provide them with iron 2. And we did see this to be the case. In each iron concentration, the wild type and pio knockouts grew comparably. And in this one, we also did not see the inhibitory role that we saw in the 4. So iron 3 doesn't seem to be inhibitory. In our ferrozene assays, it was revealed that Taiwan does indeed produce iron 3. So we expected that in the Fe2 or iron 2 chloride growth, we wouldn't see oxidation in the pio knockouts because pio is knocked out. They don't have that ability anymore. And if pio ABC is also responsible for reduction, then we also wouldn't see that in the iron 3 chloride pio knockout strains. So in the abiotic or in the iron 2 chloride, in the abiotic, we see oxidation does happen, but we grew our strains photoheterotrophically, so they were exposed to light. So this is due to photooxidation. And we use this as our baseline to think about the other behaviors. In the wild type, we see a lot more oxidation, and this is because there is actually bacteria growing in those samples, and they do have a functional pio, so oxidation of iron is able to happen as normal. In the puzzle, we see a little bit of oxidation, but because we're using abiotic as a baseline, we can say that this is because of photooxidation growing photoheterotrophically, like in the abiotic strands. In the iron 3 chloride, we see in the abiotic, we have reduction, but this is also due to photoreduction. In the wild type, we see a similar amount of reduction, but it is able, because it has a functional pio-ABC protein, it is able to oxidize that right back to iron 3. In the pio, however, we see that oxidation, or the reduction, but we see it keep going beyond what we see in the abiotic, which points towards the Taiwan pio knockout strain being able to actually reduce iron on its own. So to conclude, we know that Taiwan can reduce iron 3 into iron 2, but we cannot say that it is because of pio-ABC, because it also happens in the absence of that protein. So we answered both of our main questions. And we also found that high iron 2 may actually inhibit growth of Taiwan. So in the future, since we don't know that it's pio-ABC doing this reducing behavior, we can look for what actually is responsible for it by saying what genes are expressed when reduction is taking place, and that could point us towards the gene that's actually responsible. And we can also see what happens if we overexpress PIO-ABC to see if it maybe knocks out oxidation altogether or reduction altogether, because it's like able to oxidize much more. And then this also could be used to for bioremediation efforts. So if we know that Taiwan is able to reduce metals, this can be used towards like uranium-6, which is soluble in water and could be in drinking water and well water. But if we're able to somehow use Taiwan to study uranium-6 into uranium-4, which is non-soluble, that could participate out and have benefits for the community. That's it. And
So most chemical pathways are reversed when you sort of elute this. And that's dependent on concentration and I guess the activity of your enzyme or catalyst. So theoretically, any enzyme which can convert A to B could also convert B to A if the concentration of B is just way higher for some reason. So how would you know that, like in the environment where maybe you're on a wire or whatever, there's always a surplus of iron entering the system of a certain charge state, and so therefore you would never reach the conditions for this to work in the reverse direction. While in a sort of a steady state reaction like this, or with a very small amount of initial iron, it might be able to work the other way around. But the main question is, this process looks reversible in this reaction, and I'm saying that's because the concentration of iron is so low, and you're not able to build up very high concentration of something, which could turn the claim that this pathway turns A into D, but in reality, it turns B into A. You've just manipulated these sides of the equilibrium.
That we like originally gave them, based on what they would experience in nature. We did have a 4,000 micromolar, but they didn't seem to like that very much. They didn't grow. So I feel like any more than that would have enacted like iron poisoning. So we couldn't really try to give them more.
I see. Okay, that's yeah, that's the environmental.
I can maybe, I was the mentor on this project. You're talking about like adding maybe a mix of iron and seeing what they're doing.
Yeah, sure. That way, yeah.
That could be a follow-up experiment to this. Honestly, we were just even attempting to see if they could reduce iron at all, because this is, you're never gonna detect only one oxidation state of iron in the environment. So that could be a follow-up experiment. And it does also look like if it is doing reduction, it is certainly being buried under oxidation. But it can do it because ultimately oxidation would be it's gaining electrons, and iron 3 would be it's acting as an acceptor, and Taiwan is great because it has a lot of acceptors it can use. So a great follow-up for that.
Question. Thank you. More than that, people think. Okay, next is Ramsey, from the Fujian Lab.
Hi, good morning. My name is Rain. I'm a Vice Senior Study Economic Engineer at RPI. And this summer I've been more experienced to be in Professor John's lab with Theo to work on silk fibers. Some of you might have saw the pictures, but quick question. What kind of insect can produce a fiber that's twice as strong as steel while being more stretchable than Kevlar? Yeah, it's the spider. Another question is, what kind of animal would like to eat the male after reproduction or mating? The answer is also spiders. So spider silk is one of the materials that have very high toughness, meaning that they're very strong. At the same time, they're very stretchable. But spiders are not farmable because they are cannibalistic. So the solution to that is to produce a fiber with bacteria. The problem with that is spider silk have very high molecular weight. So bacteria usually have a very hard time trying to produce the protein. So what we're trying to investigate is if we could kind of work around that by looking at different sequences and see how that might change the property. To begin with, in the natural spider silk, it's mostly of these monomers. And with each monomers, there are crystalline and amorphous structures. And in the end, the monomers are connected into a big fiber with disulfide bond from cysteines. So if we borrow the same design mechanism as having the crystalline and amorphous region, and we kind of use amyloid and muscle protein, we can kind of create this idea of spider silk without using spider silk. So the solution that was being used is we have this artificial amyloid sequence in the middle, and in the end, we have muscle protein, and in our case specifically, the NFP5, and that's what the mechanism that's linking all the monomers together. And with all these three components, we can make this kind of recombinant fusion protein called the silk amyloid muscle protein, and I'll refer to it as CEMB. So if we have the muscle protein and we have the amyloid sequence and we combine them together, it was simply to not turn into a fiber because the self-interaction between a single MFP5 is very strong, and it's simply impossible to be turned into a fiber. So the solution we're working around with that is we cut the NFP5 in the middle, and we turn that into two halves. We attach one half at the beginning of the amyloid sequence, and we attach another half at the end of the amyloid sequence. So the one at the beginning, we call that the N-terminus, and we call it the NM-NFP. And the one at the end, it's got the C-terminus, so we call it CM. And the amyloid is literally the 16 repeats of this sequence, which have a strong tendency to form beta sheets in the anti-parallel fashion. So the question is, now we have this protein, how can we make it better? And one of the best ways to create mutations. So we're asking this question in 3 different levels is, first, if we mutate the sequence, can we have a significant change in the fiber properties? If there is a change, can we use a model to kind of understand and predict these properties? And the last is, can we kind of get to the bottom to see the chemistry or the actual mechanism behind the interactions? So previously, some efforts has been done where we mutate the CM, which is the end of the amyloid sequence, and we can see that it has very little change in the fiber properties. So we are focusing our effort on the first half, on the NM half, and we are creating a mutant library where we have a bunch of mutations in the NM half. How we do this is we create a subset of mutations. So if we are going to mutate every single amino acids in this 36 long region, that's going to be about 6 to the 10 of 46 possible permutations, and that will simply be too many to test. So the workaround with that is we kind of only mutate certain residues of this NM sequence, And we kind of also limit each change to a small range of amino acids. So these three colors means the type of change that it could possibly be, and the region kind of tells you where the mutations could possibly be. So as to kind of recap, we are changing the NMN sequence. We're expecting changes in the fiber properties. Namely, we're interested in 4, where we want the ultimate tensile strength, extensibility, toughness, and Young's modulus. And we're trying our best to control everything else. So to kind of zoom back, the method is actually a feedback loop. We call it the active learning loop. What this is we will produce mutations within each round. We make that into proteins, and we make the protein into fibers. And by mechanical testing, we will have the fiber data. And with the data, we're hoping to use machine learning and molecular dynamic simulations to kind of help us understand. And with the information, we could kind of guess a new sequence as the best. And since this is a loop, we can do this loop many, many times, and we're expected to see that the fiber properties are getting stronger. So right now we're in the first step, going from DNA to a protein. And this is the bioprocessing pipeline that we're doing, which takes about a month. How this goes is we have a DNA, we put that into bacteria, we grow them in 12 liters of media. After about 1 day, we harvest the cells, we spun them down. We use a lysis buffer, which basically breaks the cell membrane and releasing all the soluble fractions of the protein. And then we purify the protein using something called the liquid chromatography. How this is done is because in our protein, in the CMN, we have a histag, which is a long-chain of histidine, and have a high affinity to the beads. So we could kind of keep our protein while getting rid of all the junk cell protein. And with the purified protein, we are dialyzing that with all the salts within the chromatography process. We kind of freeze dry it. It's called lyophilization to get rid of these the liquid. And we resolubilize that in HFIP. We have to do this because our protein are not soluble in water, so we have to use a somewhat harsh chemical to resolubilize that. With the dope, we're able to spin the fiber, which is kind of pushing it through this dope through a 23 gauge needle into 95% methanol. And this will form a very thin fiber. And because our protein in the dope is kind of orientating in every possible way, we do this process by the post-spin draw, which kind of aligns the beta crystal, the beta sheets. And with the fiber being dried, we test it in using a tensometer. So the data that we get from the tensometer is going to be something looking like this. This is a stretching curve we got from mechanical testing. So on the x-axis, that's the strain, that is how much the fiber has stretched with respect to the original length. The y-axis is the stress in megapascal. And as I said, the 4 properties that we're measuring are Young's modulus, which is the scope of the first linear region. We measure the toughness, which is the area that's being calculated by the area by the curve. We calculate the ultimate tensile strength, which is the highest strength that it can withstand before breaking, and the sensibility, which is the highest strength that it can withstand before being stretched to flat. And to validate our data, we use ANOVA with done and post-hoc tests to see the statistical significance. So this is what the ANOVA results come back. We can see that for all the ultimate tensile strength, we see most of them have a significant change compared to a well type. And not so much in the other results, but just having this one relationship, it suffices to say that we do have a significant change in terms of fiber properties. And now we move on to the second question is can a module kind of predict the mechanical properties, which are two questions kind of stuck into one. One is how can we model the relationship? And the second question is can we produce a new sequence from this model? And how we do this, we use three different steps. The first one is featureize. What this is we're extracting meaningful information from the sequence, such as how many tyrosine it has or how many serine it has. The features can be things that are interpretable, such as biophysical and biochemical clues, but they can also be data that's fed using large data sets, such as neural network. And with the model, we can predict, kind of relate the sequence to the fiber data, and we're kind of inversing the predict model to propose a new sequence. So to kind of explain some of the methods that we use to featureize a sequence, we use CIDR and Albatross. What they are is they take in a sequence and they're going to output these properties such as hydropathy, which tells you if this sequence is hydrophobic or it's hydrophilic. There's a bunch of other properties, and Albatross is also the only properties more on the kind of conformation or the shape of the protein. Another method that we use to featureize is called Finches. What it is it is a model that uses force fields, and it's a coherent screening model that kind of models how different chains will interact with each other. And these two force fields are basically different kind of models that, for this one, it pays more attention with the pi-pi interaction. Well, pi-pi interaction is in the amino acids, especially in the aromatic ones, including histidine. It has a benzene ring and is capable of forming pi-pi interactions. And it kind of uses this as the basis to predict this epsilon score, which tells you how two sequences interact with each other. And something interesting about Finches is it can tell you how two same sequence interact with each other. In our case, this is how the NM interact, how the CM interact. But at the same time, it can also tell you how different sequences interact. In our case, it's how the NM interact with the CM. And this epsilon score is when you have a lower score, it means it's stickier, and then there's a stronger. The last featureizer we use is called Metapredict. What this is it's also taking a sequence and it's going to output this value, kind of a mean or average score of how disordered the model thinks it is. It's going to output a score between 0 and 1, and for wild type, for reference, it's about 0.915. Another thing that we tried is to kind of use large data sets. To do that, we kind of trained a neural network. However, since for our data sets, we only have 20 samples, and we couldn't fit a very good model with that. So the workaround is to use a data set that we think that uses the same mechanism. So NFP5 and liquid phase separation, we're suspecting that they're having similar driving mechanisms because they are both intrinsic disordered. And we're able to kind of intake a sequence that's proven to have liquid phase separation and output this coactivation propensity or this tendency to form coactivation. So to recap, we can take in some sequence and we can output a bunch of features, numbers, something that's representative of the sequence. And now we want to see how these features actually correlate with other properties. And to do that, we can use different validation models. So here's the heat map of all of the featureizer that we tried with the 4 properties that we calculated. And as you can see, this one has the highest. If we look at it closely, this is the disorder score versus the ultimate tensile strength. What this means is for one sample that is considered to be more ordered because it has a low score. It has the lowest ultimate tensor strength. But the other samples kind of cluster around this order region, and they seem to have this high tensor strength. And this could be useful as kind of like a safety net, so that we don't predict or we propose a sequence that we know that might have a very low property. Another interesting kind of correlation that we see is between the fitness score with the tensor strength and sensibility. So what we see here is the lower the score, meaning stronger the interaction between the two different ends, we see a increase in the fiber strength, meaning that it's stronger, and at the same time it's lower in extensibility, meaning that it can stretch less. And one of the questions that you might ask from this is, if we have three different types of interaction, how much of these actually matter within the actual fibers? And in that case, we could kind of fit a linear plane fit, where we take all the three coefficients and we fit them into a 3D Cartesian coordinate. And in the ideal case, what that would be is all the planes will intersect into a single point, and that point would tell you how much of each coefficient, how much each interaction type will matter in the real fiber. So for instance, for sensibility, we get this coefficient, which tells the story. One possible story is that you have this strong negative interaction between two different ends, and you have weak interaction between the same ends. And in some sense, it's kind of like a Velcro, where you have fabric A and fabric B. When you have fabric A and fabric A, it doesn't stick very well. Same with fabric B. But when you put these two different fibers together, they have a very strong interaction. And with this method, we can propose a sequence that Finch's think would be the strongest, essentially saying that it wants to maximize aspartic acid and glutamic acid, since they have very high disorder score, and it's acidic. So to kind of zoom back out, we walk through the process of going from DNA to protein and protein to sequence, and we collect the data and we try different models. We're gonna propose new sequences and to kind of keep this cycle going on. Here's a cool microscopy image of the fiber. This is diameter, which is 29 micron, which is very, very thin. And any questions? If no questions, here are my questions.
Great presentation. I just very curious, how did you go from your like synthesized fibers to mechanical testing? Like, how did you obtain those pressure curves?
Oh, good question. That's from this process. So we spend the fiber, we kind of draw it out. It's kind of being dried, and then we're gonna kind of take a 5 millimeter long fiber kind of clip. We're gonna load them onto this tensometer, and it's a 1 Newton load cell, so it's gonna kind of pull the fiber using these two lever arms, and then it's gonna pull all the way until the fiber is broken. And this machine will also record how much force is taken to kind of stretch the protein into each percentage, and that's how we get the stretching curves where you see a bunch of lines.
Thank you.
I think this is like a really cool project, and I really like that you are like starting with this active learning curve for scale, like stronger fibers. Did I hear you correctly that the first round you tested 20 different fiber combinations, you got some data, and then based on that plus the other extrapolate data, this model generated one sequence. Is that correct?
So technically we're really still within the first cycle. Leo has spent great effort into kind of having the first data set. I kind of just contribute to more data points. but we haven't gotten from the actual machine learning people their different methods of the new sequence. So we're only still within the third cycle. We haven't cycled it.
Okay, thank you, Ming. Let's take about 5 minutes and get a snack, get a drink, use the restroom. Ladies is this way, men's is that way. And we'll reconvene at 11:15.
Yeah, did not really like that entrance sign or not? Definitely a recommendation. Okay. There was a lot of like one of the whole thing.
I would think so.
Oh, wow. Yeah.
Yeah.
Well, I'm really going out. You're the one think what's good. Yeah. Yeah, I don't. Yeah, thanks, friends. Yeah.
Thank you.
Yeah, no, that's what I see.
let's reconvene. We have Jackson Makepeace from the Boston Lab.
I'm Jackson Makepeace. I'm a junior at Purdue studying physical engineering, and what we have been researching in the Boston Lab this summer is coacervation with the silk amyloid muscle foot protein through cosmotropic salt modulation. So just to give a brief overview on coacervation, it is a topic that has come under heavy research in the past few years towards vast biomedical and industrial applications. What it is a type of liquid-liquid phase separation wherein one or more type of macromolecule aggregates together a solution to form these protein-rich bubbles known as coacervates. And when these coacervates are isolated, they can be used to make hydrogels, biomedics, drug delivery systems, and the list just goes on. So we have been trying to form coacervates with the silk amyloid muscle foot protein. Ray touched on what that is, but it is a recombinantly produced protein. It has three major parts: the silk domain, the amyloid domain, and the muscle foot domain. The silk domain lends the protein a high mechanical strength. The muscle foot domain lends it high underwater adhesivity, and the amyloid binds them together. So these properties can be carried forward into coacervates, and from there into hydrogels or any of the applications I talked about before, and that lends them a high individual episode. That's why we're studying that. The type of coacervation we are trying to reduce involves the use of cosmotropic salts. What those are, they are types of salts that, when in solution, reduce the bulk water availability. That is the first of their two really important effects that lend to coacervation. When the bulk water availability is reduced, then there's less water for the SAM protein to stay dissolved in, so they aggregate together to stay in a liquid form because their solubility is reduced. And the second major effect is that they do something called charge screening. So most macromolecules have a net charge at pH 7. So let's say, for example, a macromolecule has a net negative charge. The positive ions from the cosmotropic salt in solution will gather around that negative charge and then allow the macromolecules to better come together because there's no repellent forces. So in past experiments with regenerated silk fibroin and literature, we found that there is a positive correlation between salt concentration and turbidity ratings, which we'll get into later. But essentially those mean that coacervation is present. And we also found, or rather this Mohammadi figure found that different types of salts affect the amount of coacervation formed differently. So as you can see, like ammonium phosphate affects coacervation a lot differently than ammonium acetate, even if the salt concentration and the protein concentrations are the same. It's also clear that at these high concentrations, or even these middle concentrations, as salt concentration increases, the turbidity values increase as well. So we started by initially hypothesizing that these effects would be the same in the SAM protein, and that salt concentration would positively correlate with turbidity readings, that salt identity would have different effects on coacervation. And we also hypothesized that different types of SAM would have different effects on coacervation. So to start this experiment, we took SAM protein synthesized from the procedure in Dowell, synthesized in Dr. Yifan Dai Lab, and dissolved these solid samples in deionized water. We then used a nanodrop spectrophotometer to estimate the molarity of these solutions, just in case all the protein did not dissolve in the water. We then diluted the stock concentration to obtain our desired concentrations. We did a very similar procedure with the salt. We took salt, dissolved in deionized water, Then with that solid concentration, we further diluted it to our design concentrations. After that, we added 100 micromolars of each solution into the wells on a 96 point and microprepared them 3 to 5 times to ensure that all wells were mixed properly. Finally, we put that in a well plate reader and we analyzed each sample individually with light microscopy. Yeah, so turbidity is essentially a measure of how many bubbles there are in a solution. So with our previous experiments with regenerated sulfide road, we were able to successfully correlate turbidity with coacervation. So as turbidity rises, the amount of coacervates insulation due to. And also just to preface the discussion later on, the main distinction between the two JL variants we're using, JL85 and JL140, is that, or one of the most important differences is that JL140 has more tyrosine present in it. So with our results, we found that salt concentration actually did not have that profound of an effect on coacervation past the initial jump you see here. So when 0.2 molar salt was added to the solution, we saw a huge jump in turbidity. So like coacervation was formed reliably there every time. But after that, as the salt concentration increased, the turbidity values either increased very slightly, completely plateaued, or even decreased, can be seen in figure 5A. So that was really interesting. And also, the 0 molar sodium sulfate concentration, or this is just like a generic salt, but 0 molar sodium sulfate had the highest amount of bubbles in solution. That did not fully correlate with our turbidity readings because we saw that at 0 molar salt concentration, the turbidity was 0, but that's not what we saw with the microscopy. So moving forward, we'll definitely have to look into that a bit more and find out why that was, but that's just an interesting result. And then as we move down the line with salt concentration, the observed coacervation solution decreased. So 0.2, it has like a nominal amount of coacervation, but as we go all the way to 1 molar salt, there's really not much to be seen under a light microscopy. So our results with material identity, especially salt identity, kind of revealed something similar. Coacervation was not that dependent on the types of ions present in solution. As can be seen here, the potassium phosphate has like a marginal lead on OD600, which really, but if up in here with the same kind of SAM, JL140 to potassium phosphate is at the bottom of each protein concentration. So we could not like reproduce these results, and so we concluded that salt identity was not like majorly influenced coacervation. However, our results with the SAM variants revealed something a little different. We found that actually did impact how coacervation, probably how many coacervates formed. We used the same amount. We used 12 micromolars of each SAM variant, and we scaled that with ammonium sulfate concentration. And we found that with the addition of 0.2 ammonium sulfate, there was a much larger jump with the JL85 than the JL140. And what else was interesting was that the JL85 actually increased, the turbidity increased with the salt concentration, which is not what we saw at all with the JL140. And then up here is a phase diagram for JL140 with 0.2 molar ammonium sulfate and 20 micromolar JL140 SAM having the highest turbidity readings. It decreased as salt concentration increased, which kind of matched with our microscopy results. This was like mildly consistent across all protein concentrations. There was a bit of an outlier with the 0.5 micromolar and 0.4 molar ammonium sulfate. We'll just have to look into that in the future. So why this was happening, we saw that salt concentration was correlated with turbidity in silk, just the silk protein. But in the SAM protein, especially the GL85, it really wasn't. So we can reasonably deduce that it's either the muscle foot domain or the amyloid domain that's causing these changes in results. And the most prevalent thing in the muscle foot domain is that there is an extremely high content of tyrosine, which is a net neutral amino acid. So in RSF is a net negative, a very net negative macromolecule at pH 7. So it needs charge screening to develop coagulation. It needs those positive ions to cover up the negative parts of the macromolecule so they can aggregate together better. But with more tyrosine added, which is a neutral amino acid, it doesn't rely on those charge screening effects as much. So with RSF, the more ions that are present in solution, the more the negative points on RSF can be cleared up. It's not the same. The muscle foot domain is the same. It doesn't need to be covered up. It can just aggregate together. As soon as this initial addition of 0.2 molar salt is added, it decreases the protein solvation enough for like mostly all protein that can coacervate and will coacervate. When the protein solvation is decreased, it doesn't rely on the amount of present solution, or that's the leading hypothesis. So moving forward with this procedure we have developed and the results we've obtained, we can pass that on to a branch of the National Science Foundation who will further our studies with high throughput screening. So what that is it's just a massive amount of like experiments. It's just a lot of experiments, 24/7. It can get data a lot faster, a lot more reliable than we can in our lab. And with that data and machine learning, we can, or they can, I guess, further explore how different sequences and factors affect coacervation, not just the amount of coacervation, but the properties as well, how strong it is, how inclusive it is, things like that. And I would like to give a special thanks to my bench professor, Lauren Gorley, Leo Penny, my PI, Dr. Flosson, and the Dr. Fuzhong Zhang at the facility.
On your graphs, there's multiple lines. Are those different variants of?
Oh yeah, it's kind of hard to read. They're the different. Yeah, sure. The green is potassium phosphate, orange is sodium sulfate, blue is
All the same protein it is.
Yes, this one is all 12 micromolar gel 40. This is 70 micromolar gel 40.
Okay, thank you. Next, we have Maddie and Henry from the Cooper Lab.
Hello everyone, my name is Maddie Henry. I'm a resident junior at the Colorado School of Mines studying electrical engineering. And this summer I worked at the lab. The Cooper Lab focuses on the development and design of responsive dynamic polymers. These polymers can be used in applications such as energy storage, health applications, and soft robotics. My particular project focused on the development or furthering the development of shape memory polymers, or SMPs. Specifically, the design and validation of a thermal cycling actuator for shape memory polymer characterization. SMPs are a certain type of polymer that you can stretch or compress, but when you apply external stimulus, such as thermal activation, it returns to its memorized shape. This is particularly valuable for applications such as soft actuators that can then be used for wearable technologies. In order to characterize how well the polymer behaves, you have to run hundreds of cycles where you let it stretch and then you apply the thermal activation to characterize it. Currently in our lab, we test that by taking a pipette of cold water and then a pipette of hot water and alternating that dose over the top of the polymer sample. As you can imagine, this makes it hard to gather large data sets, and it also introduces a lot of experimenter-driven variability. This is where my design comes in. My goal was to design a thermal cycling system that will replace the need for pipettes and instead deliver those pulses of hot water and cold water for hundreds of cycles at a user-selected frequency. Before I could begin building the system, I had to figure out what do we want for design requirements. We ended up deciding on delivery of a consistent dosage of hot and cold water at a controlled frequency, which we decided to do between a 0 and 1.8 hertz range, given the time it takes for the polymer to go back into that memorized shape. We also wanted temperature compatibility across a 0 to 90 degrees Celsius range. 90 degrees Celsius is about the max temperature where actuation will be induced for the polymers we want to work with. There's also dose repeatability. We need to be able to run consistently for hundreds of cycles where you can place a camera, observe how the polymer behaves, and you can step off to the side to perform another experiment at the same time. We also want an intuitive user interface. This is a very electrically driven system that's going to be placed in a chemical engineering lab. So it needs to be able to stand on its own and be easy to use. And we also want a single input architecture, which means you can just plug it into the wall and there's not a bunch of wiring you have to connect. That way it's more reliable in the lab set. Given that, we spent a few key design decisions. First off, we decided on peristaltic pumps. This is a certain type of pump that utilizes tubing throughout the whole system, so that is the only thing coming in contact with the water. This is particularly valuable given that most pumps have a max temperature range of about 70 degrees Celsius. But when you want the water to be 90 degrees Celsius, having that silicone tubing that goes throughout and can handle temperatures above 100 degrees Celsius makes it so it can preserve the system and reduces the risk of injury. There's also a bypass recirculation line. It's important for a stable flow to keep the system running smoothly, which is why we want the pumps to be running continuously. Given that, when the valves aren't in motion, if we didn't have that bypass line, we would be at risk for the tubing to burst, which is why we have a recirculation line that goes back into the original beaker. That way we can have a system that's able to run on its own. And then we have capped air chambers. This comes from a 2025 paper by Jang et al., where they looked at the camera peristaltic pump family, which is the same peristaltic pump we selected, and they used it to look at the dose variability with each pulse. Without an air chamber, the dose variability is about 65%, which is quite large, but when you add in a capped air chamber, it reduces down to 2.5%. And then solenoid valves are what we use for the final system. This is actually the hardest decision to make. The best type of valve for this scenario would be something called a pinch valve, which much like the peristaltic pump uses internal tubing. Unfortunately, they're mostly used for medical devices that utilize maybe a 16th inch inner diameter, which makes it unreliable for our system. It also costs about $500, which was unrealistic given that we were trying to keep the cost of an entire system under $500. We decided on solenoid valves given that they have a 20 millisecond response time, so we knew that they would reliably switch. And we also chose a single 12-volt wall adapter. This is because the pumps and valves run at 12-volt logic, but you can step down that logic to about 5 volts to successfully power a logic level converter and a TDC microcontroller. A TDC microcontroller was selected over an Arduino microcontroller, given that it has better hardware number precision, and it also has a greater impact, meaning that you can continue iterating on the system and introduce tools such as the tool computer later on. With that user-friendly interface, we want all of the electronics to be enclosed in a single PTG printed box where we'll display frequency, cycle count, and how long it's been running, and you can just move the knob to select your frequency. With that, we also needed there to be packed air tubes around. And we also needed air slits on the sides, that way the system won't overheat. For the system itself, we need to introduce something called a MOSFET driver board, because the TNC, when it signals to the valves to switch, the valves will send back voltage spikes, which will damage the TNC and cause it to burn in the long run. Unfortunately, when we were first introducing the MOSFET to the system, we couldn't get to switch the way we thought it should. The LEDs were lighting up, but we weren't observing the valves switching. What we ended up learning was that the MOSFET driver board, while it says it can run at a 3-volt frequency, you really need to use 5 volts to completely saturate the optocoupler so the system can run. Which is why we ended up studying the TT through something called a logic level converter that takes that 3.3 volt signal and sets it up to a 5 volt signal. And moving on to the fluid system itself, we will have a hot side and a cold side, and they're identical, with the only difference being that the hot beaker will be on something such as a hot plate in order to get it up to temperature. So with the hot beaker, the peristaltic pump will pull the water supply through the pump to the first T-junction where the bypass can circle back to the beaker, or it can continue down to the second T-junction where we'll have that capillary tube that will absorb those pulses and keep the water stable. We then into the solenoid valve that then connects to a line connector that we can then distribute onto the branch columns. Let's do the same thing for the cycle. The way this triggers is with the TNC as the brain. The TNC takes in the signals using code from Arduino IDE software, and it uses the rotary selector to determine frequency. It presents the cycle count and the timing on the OLED display. We also have the cold pump that runs through the 12-volt power supply, same with the hot pump. Being able to switch how fast the pump is running using a PWM line that connects to the logic. We also have something called a buck converter. This steps down the logic from 12 volts to 5 volts in order to allow the TNC to be powered on without drying it. Let me move on to the key components we want to see. So with that, we have that 0 to 1.8 hertz frequency. We have a Jones volume of 2 milliliters, which we chose due to the fact that we're still in development of student memory polymer samples. So we wanted it to be enough water that it could work on a variety of sizes. And then we move down, then up to 90, down to 0, and then a 20 millisecond response time is crucial because we need the valve to put open for no more than 540 milliseconds. And then we also utilize the TNC microcontroller because it runs at a 600 megahertz frequency, which is incredibly useful for the system because it means we can continue iterating without worrying about it not being able to handle it. And then the enclosure material is 3D printed PETG. This is due to the fact that it can handle temperatures up to about 80 degrees Celsius. So if any of our electrical components were to overheat, the system itself would still be fine. And then we decided to use a 10 position rotary selector. First off, because it's the standard to have 10 positions, but also this gives more freedom to the experimenter within the lab. And then the display is a standard OLED display, which was selected due to its low cost and the fact that we can get it even bigger by projecting it and connecting it to the computer. Moving on, we had to calibrate the dosing for the system. This was done at 150 PWM. Below 100 PWM, the system itself does not have consistent flow. And then at about 200 PWM, you start to introduce air bubbles because it's pumping too fast. 150 is about the perfect metric. Yes, there will be more water circulating back to the bypass line than at about 125 PWM, but also it creates the most stable delivery, which is the main goal. So we ran cycles of 10 pulses, and then we divided that 10 and ran 4 trials, and we found that at 500 milliseconds, we achieve 1.99 milliliters, which is ideal for our system. The R-squared value is 0.985, which is incredible for what I do, and the variability coefficient is only 1.8%, which is much below the 5% target we were aiming for. Over here we have the final system. Unfortunately, due to an EH&S safety concern, I wasn't able to get the full system shown due to not being able to enter my lab. You can see that we have the crystal pump here that goes down to the tubing, goes down to the valve, and then the Y connector. So then we distribute it over that polymer sample that would be clammed using another attachment to the ring stand. Given that this is a brand new system, we decided to develop a 6-page comprehensive user guide that goes through startup, shutdown, and then about 2 pages worth of troubleshooting given most common concerns, which will usually involve either the MOSFET board not triggering or the rotary selector becoming loose and the simple fix being just to tighten the nut. And then, unfortunately, due to the fact that the SMP samples are still in development, we only have one in the lab, so we were not able to test the system. But there is a goal to test it as early as next week once more samples are developed. And using this, the automation allows for more cycles to be completed per session compared to a manual pipetting technique. And it allows for us to further that development of SMPs. Currently, for shape memory polymers to actuate, you have to have a weight attached to the bottom of the sample. But the end goal will be to have a spring-like structure embedded within the shape memory polymer sample, so it can do the full actuation by itself without needing that weight to help with the structure. And then controlling dose, volume, cycle frequency, and valve duration is something you can do through the Arduino IDE code software that is again specified in the comprehensive user manual. This allows for the system to continue to be iterated on as the lab wants to utilize different samples. And then further work will focus on pushing more the system and using it to actually characterize the SMPs. Yeah.
Been time a concern, like the amount of time needed to let the sample reach the desired temperature?
It is not. It is incredible how quickly it is able to do what I do shape. It starts to become a concern once you're delivering water any faster than about 2.5 hertz frequency, but at 1.8 hertz frequency, that isn't a concern with the system.
These are all based on a very comprehensive design. My question is, are there any future aiming for like protecting this design like property?
At this time, I don't think so. We're going to introduce it as available equipment on the lab website. So I'm gonna have a film demo that shows how to use the system, and that way other labs can use it. But right now we don't have it as a.
Here's why you all use liquids versus maybe like a solid conductor.
Yes. This one, so the polymer samples they're developing right now are so they look like a clear hair elastic, and they were using water because that was the standard material they had in the lab. It's a fairly new lab. It's only been in for about a year and a half, so they just decided water. Good idea, and most of the literature utilizes water just because it's so easily and readily available, and it's really easy to heat, and safety wise, as long as you have tubing, you go on.
What already like exists in the scientific community for doing this sort of thing, and how does your design set itself up?
So it's a pipette of hot water and a pipette of cold water that you just alternate and you just film in it. So this one you'll also film. There will hopefully in the long run be a white background behind the system so you can actually see how it stretches and presses. But right now they tape it to the top of the lab bench using scotch tape, and they tape the pipette.
Thank you. Makayla Phillips from the Pathfinder Lab. I do want to say after this is our lunch, but the 7 students, please stick around for a moment. I want to take a picture and we have a gift for you. So don't run away until that is done. Not say we're going for lunch. Okay, let's start.
Hello, again, my name is Makayla Phillips, junior in biomedical engineering. I'm going to be senior in biomedical engineering at Purdue University, and this summer I'm continuing to the CEMB program in Dr. Fox's lab. My mechanobiological investigations span 2 areas this summer, cellular migration, as well as extracellular lipids or ECM. On the cellular side of things, we focused on hyperactive REU, which is important in cell migration, and it is also linked with cancer metastasis. So you might ask, what is REU? REU is a small family in a small protein in the GTPase family that in its active form promotes the bundling of actin fibers and the stabilization of focal adhesions. So actin filaments, they act like the cables that like transmit tension, and focal adhesions are the point of connection between the interior of the cell and the extracellular matrix. On the ECM side of things, we are focused on collagen architecture. Biologically, collagen is the most abundant protein in the body, and it plays a large role in cell migration. Things like varying collagen strain or collagen stiffness, have already extensively been studied, but varying collagen architecture has been underexplored. And this is important because in different disease states in the body, such as cancer, collagen architecture presents different properties. And so in order to create more accurate models, we need to be able to independently model collagen architecture. For my research goals for the cellular side, we wanted to capture the qualitative differences that we were seeing when observing active REU versus wild type spheroids. And we hypothesized that since active REU increases tension within the cell, it will therefore increase migration speed. For question number 2, the same side of things. We wanted to determine the effect of collagen 1 fiber width and length on single cell migration speed, with the hypothesis that increasing fiber width and length will also increase migration speed. Question 1, the cellular side of things. My lab curated a bunch of videos of active REU spheroids and wild type spheroids migrating. These are of the MCF10A cell type, which is a non-invasive breast epithelial cell line. These were all 48-hour time lapses, and in order to analyze these, I developed a SAM2 based Python software that SAM is not the like silk AMOLED, whatever they do. SAM is a deep learning model. And basically I developed this like batch processing thing that gives you a GUI and it asks you to create regions of interest over the background as well as over the spheroid. And then it uses this singular linear frame in order to create masks over all of the rest of the frames of all videos. It's kind of cool. And it's able to capture like poles, as well as like pre-flooded bodies and stuff. And then it also finds the largest contour, which is just the outline of the cell. And then in order to analyze this, I developed a tendril tracking algorithm that basically finds local maxima, and it also takes into account parameters like min distance between points in order to identify tendrils. And tendrils are just these like long extensions. And then per frame, I take the average of all the tendrils to find the general location of that spheroid, and then tracking that location across all frames, you can find the trajectory of the spheroid over time. And then you can find things like path persistence, which is the shortest distance traveled between the start and the endpoint divided by the total trajectory. And you can also find total distance. With these, we can run t-tests, and then we also ran a k-means clustering in order to find any naturally forming groups using the total mean or total distance traveled parameter. For method number 2, the collagen side of things, we plated type 1 collagen and we left it at room temperature from 0 up until 60 minutes. And this causes changes in the growth phase of the collagen. And then we incubated it for an hour to allow for cross-linking, and then we got temperature emergent architecture, or T-collagen, coined by my bench mentor Aja over there. And then we plated 41 GFP cells, which are convergenic breast epithelial cell line. These are single cells. And then we image for a 24-hour time lapse at 10 minutes. For our results, we found that active ROA does significantly increase migration speed, persistence, and distance. And just qualitatively looking at these, we can see that wild type has a lot more variability in its direction. It goes forward, it goes laterally, it goes backwards, versus active ROA is going forward. It has a more organized, more compacted structure. And we can see that at the very edges here, the edge line is a lot more pronounced. And you can see it's like 3 little like fingers that are sprouting off, versus for the wild type, it's a lot fuzzier. And then this motion is represented in our quantitative data. So with our k-means clustering, we found a threshold, and anything above that total distance threshold is considered to be regressive, meaning that its movement is forward directed. And anything below that is considered hesitant, meaning it's kind of like probing forward. And we found that wild type is majority hesitant, and you can see that with our trajectory plot, where you can see a very low persistence value, as well as a very low total distance traveled. And you see on the contrary, you see a lot of progressive samples with our active ROA data and a more persistent path and a larger total distance traveled. And this is represented by our t-tests, where we found that active ROA has a statistically larger path persistence as well as total distance traveled than wild type. This can also be seen as changing area over time, meaning that active ROA is just changing more per frame than wild type is. For our second problem, we found that altering collagen time at room temp pre-incubation does lead to significant changes in fiber width and length. And you can kind of see this when looking at our confocal images. In the negative control, there's a lot of noise. And as you move up towards 30 minutes, you begin to see more defined fibers. And then as you go forwards towards 60 minutes, they begin to become noisy again. We use CT fire in order to analyze and segment these fibers and get values like fiber width, length, straightness, angularity, etc. We focused on just width and length because we found no statistical difference in other parameters. For the fiber width and length, you can kind of see this like gentle sloping upwards as you increase in time towards 30 minutes, and then it begins to go back down again as you get towards 60 minutes. And what does this do to cell migration, you might ask? Well, we can see that path persistence and cell speed are increasing as we move towards 15 minutes, and then they go back down as we get to 30 minutes and 60 minutes. And this is represented as well in our single cell trajectory plots, where you can see that 15 minutes goes a lot further than any of our other groups. In conclusion, just to reiterate, we found that active ROA does significantly increase speed, persistence, and total distance traveled. And you might be thinking, how does that work? Because if active ROA is increasing tension in the cell, what's keeping the cell from just holding itself in one place and stagnating? So migration is facilitated by a coupling of things. And one of those things that has a relationship with ROA is RAC. And RAC basically causes the membrane to protrude outward. And so this relationship, this antagonistic relationship between RAC and RO, is what causes the cell to keep moving forward. And so if we're activating ROA, then we're probably increasing the swing of RAC as well, which is what is causing the cells to migrate faster. The other thing that we notice, the increased organization and compaction, is likely caused by the fact that ROA increases cell-to-cell adhesion. And ROA is also likely causing an increased migration of F-actin to the leading edge of the cell, which is allowing the cell to put more tension down. And then these formation of defined philopodium allow for more localized force distributions and a harder pull, allowing cells to go faster. For the second part of the project, we found that collagen fiber width and length trend towards a biphasic relationship with increasing time at room temperature. What this increasing time at room temperature is likely doing is, I'll roll back a bit. The way collagen is formed is you have the nucleation phase, where you have these collagen seeds, and then you have a growth phase where the collagen seeds grow in width, they grow in length, and then you begin to plateau and the collagen becomes more compact. Time at room temperature is impacting the growth phase, so it's allowing these cells to have more time to grow, or allowing the collagen fibers to become wider and longer. We're likely seeing this upward movement and then drop off because the collagen is probably starting to degrade as we get to a certain time point. And then the single cell migration speed peaking on 15 minutes is likely not caused by fiber width or fiber length, but instead by an increased access to binding sites on the collagen as we move towards 15 minutes before we begin to degrade pastwards once we get to 30 minutes and 60 minutes. For future work, we would like to investigate cell viability of T collagen, analyze the T collagen pore size, because if we plate the cells in like 3D collagen, literature has found that smaller pore sizes cause the cells to move into an amoeboid state with basic microarray. So that would be interesting to look at. Yeah. Oh yeah, and improve the spheroid analysis pipeline. Because sometimes SAM makes mistakes and it thinks that the background is on interest. And so one way that we can circumvent this is by making a clause that the mask chosen has to be the mask with the highest grayscale variability. Because we can see that there's like a lot of brighter areas and lower areas within the sphere body. The other thing is sometimes the tendril tracking algorithm will mistake tendrils. And so instead we could just take the average of all points along the contour. And that could be the point represented in point perfect. I would like to thank my lab. I had a great summer with them, and also the funding source, Anisah. Thanks. So, yeah.
In your method section, you have this sort of 8 sample. Is that the throughput of the video measurement?
So these two are two completely separate projects. And this is not representative of what our wells look like. Our wells are actually 6 well plates, but in order to fit everything into one figure, I just made it into an 8 well plate because we created 2 6 well plates. And so yeah, the videos are for the spheroid analysis. This is for the single cell.
I see. And so there you were using a 6 well plate with the microscope. Okay.
Is that a question about potential impact of this research? Is this understanding of the basic this impact inhibits the growth so this can't spread? Is that the end target? Just saying.
That's possible. Yeah, a better understanding of how cancer metastasizes could lead to future therapeutic development. And then for the second project, being able to better model like physiological microenvironments can allow us to more accurately study how cells would behave in those cancer.
Good, thank you. And this concludes our presentations. Thanks to everyone for your attention and for being here, your great questions. Thanks to your terrific work, and the labs for your support, and our BAM managers for also supporting our students. So it's been a great summer. We'll miss you, and safe travels back home to all your students. Again, let's have the students come up to the front. I'm going to close the screen and lunch have on here for our lunches. Yes, indeed. So the lunch is in Green Hall. That's the next building that way on the 2nd floor. So I'm going to go down 1 level on these big stairs. And that way, kind of around the corner, there's a gathering space there for lunch. Ready for hungry? You pick up the snacks on the way out. Well, and thanks again. We'll see you at lunch. Students, come on up.
Hey.
Go. Welcome back this year.
And if there's anything in this slide file that you want, that's but the third
น้องหันมา
Ellie in the front, Anisah,
Wait, you want us over?
and then Anisah on the other side. Makayla. Oh, yeah, you got it. You got it.
Well, we are.
Are you in there?
Okay, please discuss about.
Okay, okay.
They just travel back that way.
Right.
Yeah.
Probably doing like a three-way.
Yeah.
Oh my.
Yeah. Yeah.
Guys, here, I'll help you get your.
All of that, we are.
Right. Oh wait, I said yours eight.
Maybe like.